Fingerprints are a distinct pattern of ridges and valleys found on the pads of our fingers, classified into three main types: loops, whorls, and arches. Remarkably, no two fingerprints are exactly alike, even among identical twins, making them an essential tool for identification. These patterns begin to form in the womb and are influenced by both genetic factors and environmental conditions during fetal development, which means they remain unchanged throughout a person's life. In forensic science, fingerprints play a critical role in criminal investigation, helping to accurately identify suspects and victims. Law enforcement agencies utilize techniques like fingerprint lifting and digital scanning to collect and analyze prints. Additionally, fingerprints are increasingly used in security systems such as smartphone and biometric access controls, providing an extra layer of safety. Interestingly, the concept of unique identifiers isn't limited to humans. Animals like koalas also have distinct fingerprints, so much so that they can confuse forensic scientists. The study of fingerprints, known as dactyloscopy, has a rich history dating back to ancient and has evolved significantly with advancements in technology. From criminal justice to personal security, fingerprints serve as a powerful reminder of our individuality and the intricate complexity of human biology. For blind people, fingerprints serve as a critical means of identification, especially in environments where visual recognition isn't possible. Just like sighted individuals use visual cues to recognize faces, blind individuals rely on their fingerprints to tactile memory. Fingerprints are formed during fetal development between the 10th to 16th week of pregnancy. The patterns arise from the interaction of genetic and environmental factors. As the fetus grows, the pressure and the movement of the developing fingers against the surrounding amniotic fluid creates unique rigid patterns, which are determined by both genetics and random environmental influences. The outer layer of skin, known as the epidermis, contains ridges that form our fingerprints. Beneath the epidermis is the dermis, which contains sweat glands and other structures. The friction ridges on our finger enhance our ability to grip surfaces, providing better tactile feedback, which is especially important for tasks requiring precision. Blind individuals often develop heightened tactile sensitivity, which allows them to interpret textures and patterns more accurately than sighted individuals. Studies have shown that blind individuals can be more adapted at recognizing their own fingerprints through touch, demonstrating the brain's ability to adapt and enhance sensory processing. Neurological aspects. The brain areas responsible for processing touches and spatial awareness may become more developed in blind individuals, contributing to their ability to rely on tactile information. This neuroplasticity showcases the brain's remarkable ability to adapt to sensory loss, allowing blind individuals to engage with their environment in unique ways. In conclusion, fingerprints are not just a means of identification. They represent a complex interplay of biology, genetics, and sensory prescription. For blind individuals, their fingerprints provide both identity and critical tools for interaction with the world. These are fingerprints that I have done of myself, and they would not let me record, but I did get fingerprinted taken of myself, and this would be the way they would do it for fingerprint identification.